Hi, everyone. Welcome to the AGM Preclinical Translational Science webinar. For those who don't know me, I'm Serene Markosian. I lead the Assay Guidance Manual program here at the National Center for Advancing Translational Sciences. Today's speaker is Dr. Rommie Amaro. Rommie holds the Distinguished Professorship in Theoretical and Computational Chemistry at the University of California, San Diego. She grew up on the south side of Chicago and received her BS in Chemical Engineering and her PhD in Chemistry from the University of Illinois at Urbana-Champaign. She then did a NIH postdoctoral fellowship with Professor J. Andrew McCammon at UC San Diego, and then started his independent lab at the University of California, Irvine in 2009. In 2011, she moved her lab to UC San Diego. She is the recipient of multiple awards, including the NIH New Innovator Award, the Presidential Early Career Award for Scientists and Engineers, the American Chemical Society Comp OpenEye Outstanding Junior Faculty Award, and the American Chemical Society Kavli Foundation Emerging Leader in Chemistry Award. She also received the 2020 ACM Gordon Bell Special Prize for COVID-19. Rommie's scientific interests lie at the intersection of computer-aided drug discovery and biophysical simulation. Today, she will be talking to us about her award-winning work on in situ computational microscopy of SARS-CoV-2 and her thoughts on challenges regarding the utility of AI/ML in drug discovery. I got to know Rommie recently as we worked together to build this amazing workshop called In Silico Drug Discovery Workshop that is going to happen virtually in October 23rd, 24th, so very soon. And if you have not registered yet, we're going to drop the link to register on in the messages. So go ahead and register. This is going to be a phenomenal upcoming workshop. One more note before we start. Questions, if you have any questions, please place them in the Q&A box and we will get to the questions at the end of the talk. And you can always upvote questions if you think some of these questions are interesting and you'd like to hear the answers. So with this, on behalf of the AGM program, my colleagues here at NCATS and everyone here listening today, we're very excited to have you, Rommie. So, and we're looking forward to your presentation. The floor is yours.
Let me make sure I'm in the right PowerPoint. Thank you so much for that really nice introduction. I'm really happy to be here today. I'm Rommie. So good morning. I think it's still morning in most of the time zones. Right. So today I'm going to be talking about a mix of things. And I just want to say up front that I know the title of my talk is Artificial Intelligence and Machine Learning in Drug Discovery. And what I'm going to talk about is closely related to that, but on reflection, I think maybe a slightly better title would probably be something like AI and ML in biological discovery and in discovery of new biology. And I'm going to use the story of our work in SARS-CoV-2 to sort of paint a picture of where things are at, especially in the space of molecular biology and molecular simulation. But we'll just go ahead and get started. So the past few years, I've been serving on an advisory board for the Office of Advanced Cyber Infrastructure at the National Science Foundation. And as part of that, participated in a working group that was sort of really focused on trying to identify what are the opportunities in AI and machine learning. This was back 3 years ago now. But I think in the space of biology, while we've had an enormous number of advancements since then, I think nowadays they feel like they're coming just so rapidly. There are still a lot of the challenges that we identified several years ago are still with us today, and I think they're going to probably continue to be with us maybe over the next decade or so as we really sort of change our practices. And one of the things that was that came through in this work group, which is, I think, to me, one of the most important aspects of being in this space, of working in this space of computational biology and chemistry, is that there really has to be take full advantage of the opportunities in AI and ML, whether it's drug discovery or biologics discovery or just science, AI for science, there really has to be a very close coupling between those who are advancing and developing new computing technology and those who are utilizing or using those methods and platforms to actually do the science, engineering, and discovery. And this has always held true. And I know, at least in my career, we've always been a group. My lab has always been a group that sort of really worked at that interface of sort of, sort of interesting biological questions at the place where, you were really pushing the use of new supercomputers or different types of algorithms. And we've been in that space for a long time, but this feels now truer to me than ever before. And just a few comments. So I sort of begin and end my talk with comments, and then in the middle, it's sort of interspersed with all the science and some of the methods. I think that probably for most of us, maybe you feel, at least I do, I feel like we're living through a disruptive moment where we have developments in ML and AI coming at really an extraordinary pace. And I think myself and many others believe that AI and ML in biology is going to be huge, but this is not something that's going to happen overnight. Part of the issue with that is that current biological research is in a lot of places, it's still very sort of artisanal, if you will. And, as others have said, we're sort of in a pre-industrial revolution state in terms of our methods, our data, how we really share them, just sort of like the whole gamut. And I think that in order to really, take full advantage of the opportunities in this space, we have to transform actually many aspects of our research practice and research process, from all the way from how we even think about biological problems, where data is not just an outcome, but it's actually a key driver in a lot of these strategies, how we actually execute the research, what we do with our data, how we connect with other fields, and even including how we publish. I mean, our ways of publishing are just really kind of abysmal if we want to advance rapidly in this space. So, there's a lot of people trying to tackle different elements of this, and I think we need to sort of group up and try to meet this opportunity, meet this moment. So I think that the work that I'm going to tell you about sort of paints a picture that tried to address a lot of these challenges in real time, but during the SARS-CoV-2 pandemic, especially the early days in the first 1 or 2 years. And I think that was critical because we were, on one hand, of course, it was a terrible thing, with millions of people dying, and it was just, from a public health perspective, really just awful. But there was a bit of a silver lining in terms of, some of the science, and in terms of people coming together and sort of shedding our preconceived notions about how science should be done, and in order to really sort of answer a question. And I think that gave a wonderful opportunity to do things that otherwise would not be possible, and even today are really no longer possible. That there was a window of time, and that's the window I'm going to talk to you about. And then it's sort of closed. I mean, it's still there a little bit, but it's not the same as it was. So in order to, I think, really to tell this story, I wanted to start with the team. I mean, this is a scientific story that involved a huge number of people, both that covered sort of that gamut of what I talked about, from the people actually developing the computers, so the folks at Summit, which is the big compute at Oak Ridge National Lab, also at the Texas Advanced Compute Center, such as Frontera and so forth. We worked with folks directly at NVIDIA who were, really keen to port new algorithms, to these platforms and to their chips. And then we were working directly, of course, with biologists who were giving us their data, before it was published, and like really sharing and so forth. So it was a really very well networked team that covered all these different aspects that I think are part of the modern enterprise for trying to utilize and take advantage of machine learning in this space. And that was in 2020. And then in 2021, we actually took it to another level where we expanded a team of about 35 to then to a team of about 60, and even brought in a few more different types of AI methods and algorithms in order to to try to accelerate discovery in this space and sort of, take advantage of sort of a wealth of resources that were made available at that time in order to advance scientific discovery around SARS-2. So this is, I mean, I can't go through all their names, but it was really just a fantastic experience. The other thing that, so teamwork central, and where you're really partnering, discovery scientists, sort of domain scientists with compute and algorithm people. But the other aspect was really about data and data sharing. And one of the things also that sort of was unique in that time was the attitude that scientists broadly adopted, that we were going to, very rapidly make available the data that we generated. And what this did was catalyze, a whole amazing series of discoveries that allowed us to elucidate the fundamental biological mechanisms of this virus in a pace that it was just really unparalleled almost for scientific discovery until that point. And this is just a couple of examples here, even in the molecular simulation community, which arguably has been a bit behind in terms of developing fair data practices and sharing of methods and models. Even we, because that's part of my community, and it's a mindset that, myself and others have been really trying to work to change. But even this community, which is ordinarily is more sheltered, even said, yes, we're going to just share everything openly. And we did that. And it was really also part of what led us or enabled us to do this work in sort of short order. Okay, and so what's this work that we're talking about? Well, what we really wanted to do here, and sorry, I feel like this is a big busy slide, but what we were really interested to do was understand the biology of the virus, and in particular understand what happens, understand its mechanism of infection in terms of the spike protein, which I'll tell you about, as well as to understand what happens to the virus when it's aerosolized. And, part of one of the big challenges with COVID is that just simply washing your hands and disinfecting surfaces is not going to stop transmission. And that's because this is an airborne pathogen. And so what happens is that you have droplets, which are large particles that are expelled when you talk or sing or breathe, but you also are releasing these smaller particles called aerosols. And droplets will actually, go about 6 feet, and then because of their size, they drop, which is kind of how we got to that 6 feet rule for social distancing. But what we didn't appreciate early on, but that I think, and we haven't done as good of a job as we need to addressing, is that, of course, the virus actually gets enriched in these smaller particles that are emitted from people when they breathe, sing, and talk. And these are called aerosols. They are small, and because they're small, they float. They can float very long distances, and they can stay in the air for hours and still be infectious. And also that changes with conditions such as high carbon dioxide. So when you're in these poorly ventilated indoor spaces, you end up with a much higher chance of getting COVID. In any case, there are tons of scientific questions, like trying to understand what's happening at each of these scales, and each of them being interesting. And I think it ties back to the core central idea that biology is really multiscale. You can't answer any biological question. You can't really answer it with just one technique. You need many different techniques. And so, so what we were trying to do was bring together different types of experimental data sets across a diversity of scales, from the atomic and molecular, all the way through to cellular, and even, in the aerosol type continuum of spatial and temporal scales, to bring them together to try with physics-based simulation to actually understand what's happening. And so I personally think that this challenge of understanding biology in general is really multiscale, multimodal, and it takes an integration of domain sort of areas to really answer. And I think a wonderful question to sort of bring this approach together is really with airborne transmission of disease, in this case SARS-2. Okay, so what I'm primarily going to focus on are how we were using a technique called molecular dynamic simulations as coupled in several instances with AI algorithms and ML algorithms, which I'll tell you about, in order to gain new insights into the virus dynamics and structure, into its infection mechanism and its structural dynamics in situ. And so I like to think of this method as a computational microscope because that's really sort of what it is. What we can do nowadays is combine many different types of experimental data sets. So this could be different structural data sets taken at different resolutions. So you can imagine high resolution X-ray crystallography, you could have single particle cryo-EM, you could have now cryo-electron tomography studies. And we can bring those different data sets together and then augment them with glycomics data, with lipidomics and genomics to build these highly detailed three-dimensional physical models of biological systems. And in this case, we're really focused on the SARS-CoV-2 virus at different scales, as I'll tell you about. And then all we're doing is basically, we build this structural model, we then approximate that system down to its many atoms, okay? And this could go from having tens of thousands of atoms if you're studying a small part of the system to actually having over 1 billion atoms. And so so spanning spatial scales now from angstroms to micron, and from temporal scales basically from pico and nanoseconds to sort of like small detailed molecular behavior out through seconds time scale type dynamics. We approximate the system down to its many atoms, and then we define this potential function here. And I have to say, this is the only set of equations I think I'm going to show you. I'm pretty sure I took the rest of them out. I work actually pretty hard to take the equations out of my talk. Even though this is an AI and ML talk, maybe I shouldn't have, but in any case, we basically define this potential function, which describes the different interactions that each atom has with all the other atoms in the system. And we rely on these things called force fields, which basically feeds us parameters that have been trained on experimental data sets that are used together with this equation and Newton's equation of motion, as you can see here on the right. And basically what we do is we start with a particular configuration that's derived heavily from experimental data sets. And then we integrate a time step. And this time step is about 1 to 2 or 4 femtoseconds, depending on sort of the algorithms that we're using. And then we get another structure. We integrate again, we get another structure. We do this numerical integration millions, billions, trillions of times, and basically what we then end up with is sort of a dynamical trajectory of the system's movements over time. And that information tells us, as I'll show you, all manner of things about sort of the dynamics and the function of these like amazing molecular machines. And so nowadays too, and I'll just mention this, I probably won't have time to come back to it at the end of my talk, but all of the science in this space has been dramatically accelerated thanks to, for the most part, graphical processing units. And the same technologies that, initially were used to drive the billion-dollar gaming industry, mainly by NVIDIA, it turns out that the types of calculations that I'm talking about here actually map really well onto those chips. And so there was like hand-over-hand accelerations in terms of the capabilities of these methods to actually answer questions that are of interest to real biologists, as opposed to where they had normally been, which is sort of like smaller questions in chemistry and the chemical sciences and more model systems. So in any case, we utilize some of the world's fastest and best supercomputers that exist here in the United States. One is called Frontera, that's at the Texas Advanced Compute Center, a fantastic team of folks there. And then another machine called Summit, which is at Oak Ridge National Labs, one of the DOE machines. Okay, and so all of us by now are really familiar with SARS-2. This is basically what it looks like. It looks like a golf ball with spikes sticking out of it. Those spikes are really important because they sit on the outside of the virus, as you can really clearly see. And so they're the first point of contact that the virus has with human cells. So they play a really key role in the infection process. And on top of that, these spike molecules are also highly immunogenic. And so they contain epitopes that our body responds to in order to protect from, using mechanisms of adaptive immunity, and are part of pretty much all the available vaccines that we have available to us. So one of the main things that we initially wanted to focus on was trying to understand that initial infection event. And this is described as shown here in the pictures, where you have the spike protein, which is shown here in white with little blue guys coming off of them. I'll talk to you about those blue guys. Those are glycans. And it's making, what it has to do is basically make contact or find on the surface of the host cell a molecule called ACE2, or angiotensin converting enzyme 2. A zoom in of this interaction is shown here. And so basically what happens in the first step of infection is that the spike protein and this little domain here highlighted in light blue, it's called the receptor binding domain, makes contact with the receptor on the host cell, which is called ACE2. And it's just this amazing, relatively sort of like small surface contact that, you know, really sort of drives the whole set of downstream events in infection. So, you know, one of the things that happened very rapidly was structural determination work. There was an initial paper by Jason McClellan and collaborators at the National Institutes of Health that provided the first high-resolution view into the spike protein that's shown here on the right. This paper was pre-printed into the bioRxiv on Valentine's Day in 2020. It was published 1 month later, as you can see here in Science, and has already been cited, I think, like over 15,000 times. Really, really critical information. It gave us the sort of, you know, the atomic positions of the spike head, which are critically important to know. And it had the receptor binding domain that that's really important for infection in what they call a one-up conformation. A few weeks later, another fantastic structural biology group, the group of David Bietzler at the University of Washington, published a second study that largely recapitulated the finding of Jason and his team in terms of the one-up open state, but they also used symmetry to create a model of the closed state of the spike. And so one of the things that happens, of course, when in structural biology is that, you know, they try for very high resolution structure, but there's always things that are moving a lot, things that are highly dynamic, you know, they're sort of recalcitrant to structural characterization. So we can use computational methods to basically build in missing segments. We did that. We built in various missing loops that you know, particular protonation states, because these things don't have hydrogens and so forth. And that kind of, those sort of details are really important to the function and dynamics. And we modeled all of those. Now, we wanted to go beyond just looking at the head because we wanted to really sort of understand the full structure. And so we also made models of the stalk protein. And actually, this was pre-AlphaFold, but this is a place, of course, where AI has been really, I think one of the biggest splashes in biology, of course, has been AlphaFold, AlphaFold 1, AlphaFold 2, AlphaFold 3, in terms of being able to provide pretty highly correct, in most cases, structures for proteins that have not been otherwise experimentally structurally characterized. At the time when we did this model, we used other methods that, you know, are regression trained and so forth. So they're sort of machine learning like. But that is a place where now, of course, there's a lot of there's a lot of continued interest in continuing to refine and develop those models. But in any case, these methods allowed us to basically develop the rest of the domains of the spike protein, which they could not actually structurally characterize at high resolution with any experimental technique. And that's just because they move too much, essentially. So we were able to build in the stalk and the transmembrane component. And then the other thing that they can't structurally characterize yet, experimentally at least, for the most part, are N and O-link glycans. And I think as probably most of you know, these are a very important post-translational modification of proteins. And for viral proteins, they take on they have additional significance because basically you have these oligosaccharides that are added onto some asparagines and threonines that and serines that basically can essentially hide these spike proteins or the viral glycoproteins from the human immune system. And so although experimentalists can't characterize structurally what they look like, we can use glycomics methods or different mass spectrometry to actually determine what is the molecular recipe at each of these different sites. And then we can build those out computationally. And that's exactly what we did. So we got the recipe from Max Crispin and coworkers and Paris Duazati of like what the propensity was for this glycan to have this particular occupancy and this particular composition at all these different sites. And then we modeled and simulated those. And so this is basically what this simulation looks like of the spike protein. We have the entire the entire end-to-end distance of the spike protein. We have it in the up state and the closed state, and we have all of these different glycans. You can see them sort of moving around. And one of the things that I want to emphasize in this talk, the sort of interplay between machine learning and AI with simulation. One of the really great things about simulation is that it has allowed us, just even sort of before we get to the point of AI and ML, but it's complementary to it, is that these simulations are based on rigorous theoretical principles of statistical mechanics. And so that means that, you know, when we're looking at these atomic level motions, we're actually using physics to predict what they're, based on sort of a set of information initially, what their future movement is going to be. And so this sort of like, it really allows us to sort of make direct connection with experiment in terms of linking time averaged microscopic properties that we derive out of these simulations to experimentally testable macroscopic observables. These are things like binding free energies, heat capacities, all manner of things can be sort of like directly linked. So it's really sort of a powerful approach that allows us to extrapolate pretty far beyond where experiments and the data was previously. And I think that's one of the key things that the interplay with AI and ML is like, really, can we learn new things? How do we go beyond the training set data? And I think that to me is just going to come from, and I think, you know, I'm not the only one who thinks this, really sort of the blending of these data centric, you know, ML and AI methods, but together with simulation, different simulation types in order to learn new things and to go beyond, you know, sort of to really sort of take full advantage of the opportunity. So, so what did we learn? Well, one of the things that these simulations showed is that, you know, we actually showed what the spike protein actually looked like. So on the left is what the sort of the structural biologists see. You see the protein, and that's because those glycans, as you saw in that movie that I just showed you, they're really moving around. They're highly dynamic. And so, as I mentioned earlier, these dynamic regions of structure, you know, they're hard to get a crisp picture of or to resolve using these experimental methods. So that's what the experimentalists see is sort of on the left, and what we see is really on the right. This is actually what the spike protein actually looked like. If they could actually see the glycans, which were there, but they were just too low res, they would actually see, we would have seen what the spike looked like on the right. And you can basically see that each of those tufts, the dark blue sort of puff balls here, this is a composite image of basically one end-link glycan and sort of the space that it has carved out in the simulation. And so what you can see is how effectively that these glycans basically shield the spike protein surface from the immune system. And this is the so-called glycan shield. And this is, you know, critical information really for vaccine discovery and design, and also for drug design. One of the things that was interesting about this was it really showed very clearly why the spike protein had those different conformational states. So it has to go into the 1-up conformation, because otherwise this receptor binding domain, which I'm showing here in blue, so on the left is the closed spike, on the right is the open spike. When this is in the all down or closed conformation, this region that needs to interact with ACE2 in order to precipitate infection is completely shielded. And you can see that from the side, it's sort of tucked down underneath the glycan shield. But in the 1-up state, it presents a completely different surface. And actually this part of the RBD that needs to make that handshake with ACE2 is actually, you know, exposed and ready for binding. And in addition to showing just the shielding, which people knew about already, that these glycans would shield the surface, one of the really interesting things that came out of the simulation was that we also discovered on working with the structure and looking at the dynamics, one of the things that jumped out to us is that there were two glycans at position 165 and 234 shown here in the middle in sort of in space filling representation. When we rebuilt the glycan side chains, what we saw was that the the side chains of these glycans actually filled up the void space that was created when the RBD went into that up conformation. And so it led us to the hypothesis that these glycans might be playing a structural role, helping to prop that RBD up into the open conformation. And in fact, this was tested experimentally by our collaborators, Jason McClellan, and was really neat because it actually showed a new role, a new sort of biological role for glycans, which was that not only are they shielding, which everybody knew about, that they're shielding these viral proteins, but actually that they have the ability to play a key role in the infection process. And so, okay, so that was sort of, what we did with sort of baseline simulations, looking at the sort of conformational landscape around these native sort of like energy minima. But one of the things that we can now do with simulation too is not just look at sort of the low-lying states around the closed state and the low-lying states around the open state or different conformations, we can actually look at the opening process. Now, the challenge for simulation here is that the opening process of the spike, we believe it happens it's about on the order of seconds. This was shown using FRET experiments by the group of Walter Moffat at Yale. And so, that's to get to the seconds timescale with simulation is pretty prohibitive if just using conventional methods. So we partnered with Lillian Tong and coworkers to actually apply what we call an enhanced sampling approach, which is just a way of maintaining the theoretical rigor of the simulation so that you can still connect to experimental observables, but it sort of dramatically accelerates what you can actually sample. And so what I'm showing here, what that means is that we started with the closed spike, and then over just a few weeks of compute time, we were able to actually simulate the opening, and you'll see it here going from the closed to open transition. It's about to open, some movements happening, and then it opens. And one of the cool things about this is that it's very efficient as the energy barriers get harder to go over, you get actually more efficiency from these methods. So what that allowed us to do was basically look at the opening. Now, going back again to sort of machine learning and AI methods, one of the areas where folks have been really interested to apply them is, again, in structural biology in a number of different ways. One of the things that structural biologists have sort of focused on in the past, of course, is, they're very concerned with getting that sort of high resolution structure, particularly for X-ray and for single particle cryo-EM. They get like that one high resolution structure, you publish a science paper, and then you sort of move on. But when en route to getting that structure, there's actually a lot of data that they don't necessarily take advantage of. And these are states that, these are basically, there's states that are sampled that are at lower resolution that are there, but they sort of essentially ignore. So there's a lot of folks trying to take better advantage of those data sets and try to use them to understand sort of beyond just that high resolution state, what are some of the other metastable states that might be sampled? And one ML-based method that was developed by Joachim Frank and some folks at Wisconsin is called manifold EM, where they basically can sort of take all of the data, learn the underlying manifold, and try to connect that to the energy landscape. So coincidentally, it turned out that, well, the whole world had been basically working on Jason's 6 VSB structure. And so they had been so we simulated that structure and did the opening trajectories. And Evan and Joachim Frank from Columbia had been trying to apply their manifold EM method to that same data set. And because of limitations in that data set, it wasn't very complete, et cetera. They couldn't they didn't have a very high resolution or sort of a very excellent way on their own to sort of publish their data applying manifold EM. But what we were able to do was actually extract out from their, using their analysis, we could basically we actually found opening pathways. And so I'll show you here. So on the left is the weighted ensemble trajectory showing that opening motion. You can see it goes from the closed to open. This is actually from our simulations. In the middle panel and the right, and I'm going to play that again, you actually, the middle panel and the right are actually the manifold EM pathways that they found. And so on the left, again, is the simulation data. On the middle and the right, I'll play it one last time, is data that's actually extracted from the cryo-EM data that was like not actually characterized in any study until here. And so what is it telling us? This allowed us to find sort of this weak signal, if you will, of the opening in the experimental data, and then compare it to the trajectories, these opening trajectories that we had developed using simulation. And what you can see is a very high alignment with, or agreement between, the predicted opening structures and trajectories, if you will, and the experimental data. So what this did was give us extra confidence that the pathways that we sampled using simulation actually were possibly actually biologically relevant. And one of the interesting things that fell out of this study where we looked at the mechanism was there was a new glycan, which I'm showing here in hot pink, which is at position 343, which when we just did our conventional simulations looking at the closed state and the one-up state, if you look at this glycan, all you see, it's just hanging in the closed state, it's just hanging out at the side. It doesn't look like it's doing anything interesting. In the one-up state, it's the same. It's down here at the side. It doesn't look like it's doing anything interesting. It's not until you actually look at the mechanism of opening that you see that this glycan is actually a very active participant in opening that receptor binding domain. It's making a number of contacts with these different residues, and then at one point basically acts almost like a crowbar to lift it up and out. And so they went back, Jason and colleagues went back and did mutations and functional analysis on that. And they found this glycan is actually even more important than the first two that we found that were sort of like structurally important. This was actually important from the opening mechanism standpoint. And actually, if you get rid of the glycan here, the spike basically can't even infect cells. Okay. And I know I'm going to try to cover a lot of scales in like the last 7 minutes so that we can have a good amount of time for discussion. But at the next scale, we were really, again, interested in understanding what's happening with the infection mechanism. So we built an even larger scale system that allowed us to bring together these two proteins. So this is the spike protein together with ACE2, and we have our two membranes. And here you can sort of see the dynamics of these two across the membranes. And yes, this is simplified, it's reductionist, it's missing quite a bit of the, actual cellular components, but we have this essential interaction. One of the challenges with simulation as you go to larger spatial scales is that it becomes computationally more demanding to actually study or, to reach time points. They're slower basically. So Arvind Ramanathan and his team, he's at the Argonne National Laboratory, have been working on a method that they call Deep Drive MD, which uses, again, it's a machine learning based method that uses deep learning in order to take structures that have been generated at the smaller scales where we can do pretty aggressive sampling and then use them, learn on these, and actually use them in the interpretation and the driving of simulations at the next larger scale. And so they can build these physically interpretable embeddings that allow us to identify interesting conformations in sort of the phase space that the molecule is experiencing, and then revisit those in sort of an iterative way to basically to try to tease out more interesting elements of the conformational landscape and dynamics that are of these spike proteins in these more complicated scenes. And I'll just note that these folks have just recently, I think just last week, actually published a nice they put another preprint in sort of on this method and its application to protein folding. So I think that's been a really exciting development in terms of using machine learning and AI to make these simulations faster, because I think that's again, one of the big limitations is sort of the time to compute and to discovery, where AI can really accelerate that. So beyond just the next sort of scale in terms of multi-scale that we wanted to really understand is to take advantage of cryo-electron tomography data, where now they actually they can actually look at viruses infecting cells in situ, and together with focused ion beam milling can actually get these very high resolution pictures of like what's happening as the virus infects cells. And so this is sort of the output of that, is that they can understand, you know, sort of the average size of the viruses, the population of the spike proteins, and even the conformation of the spike proteins. And so we get these sort of molecular maps that we can use to rebuild models now using our all atom approaches, but at the next level. And so, you know, we build the membrane, we have our spike proteins now embedded in here. We also have the E pentamer. There's this E pentamer that's in there, and also the M dimer, which is more of a structural protein that plays with the nucleic acids a bit on the inside. We have all of that. And then what we can do is actually stimulate this larger scale virus at this scale. So this is now getting to about 305 million atoms. And it has, you know, it's the dimensions of sort of, you know, what they see it's the actual dimensions of the virus. So it has about a membrane that's about 90 nanometers in diameter with the spikes coming off of it. And so there's many things that one can imagine doing with that virus, you know, and things that we're working on now are sort of like trying to build host cells and try to really understand how the virus is interacting with things like glycosaminoglycans and so forth, the glycocalyx and the whole cell surface. But we were really interested here to understand what happens to the virus when it is in these smaller aerosol particles that, you know, that basically float in the air for hours and can be inhaled by others. And so there's a number of different types of data that basically tell us what are the molecular components of these aerosols. We have the virus inside of these aerosols now, and all these different components. There's things from like from your lung fluid, for example. You have albumin, DPPC, DPPG. You have mucins, cholesterol, all manner of ions. These are now in here, and as part of this, we also developed these first-in-class mucin simulations. Mucins are amazing. I have to tell you, I am so fascinated by glycobiology, and I think it's one of the most, I think it's just a super, super exciting area. In any case, we built these glycans, which are basically these sort of like long peptide chains that are decorated with O-glycans. And of course, they are really difficult to structurally characterize. There's some really cool imaging now that's been carried out by the lab of Henrik Clausen. And they actually align very well with the simulations and sort of the structures that we're seeing coming out of them. And so we have a preprint in the archive about that. But what we were able to do then is to really bring all of these components together and actually then simulate their atomic dynamics, but like in the aerosol. And so here's an example. If you squint and you get your eyes close to the screen, you can actually see that all those little molecules in there are actually moving. And so they're allowing us to understand the interactions of all of these different components and the virus. As you can see inside, we can actually start to understand what's happening in situ. And this has now 1 billion atoms and is about half a micron or so in size. And here's a zoom in, you know, of just looking at sort of like one spike protein, these different interactions that are happening inside of the aerosol according to these predicted models. And again, one of the things that we were really interested in that is falling out of these simulations is that it appears that mucins are actually sort of sticking to the virus, the virus has become over time very sort of electropositive. Especially the spike protein actually has a very highly positive surface, which actually accelerates and strengthens the interactions of the spike proteins with the glycocalyx. But in the aerosol phase, what we're also seeing is that there's the interactions with mucins, which are also very negatively charged or highly enriched. And not only that, but these interactions are sort of mediated by calcium ions. And so they sort of make this sort of cross-linking type, almost gel around the spike proteins and the virus. Another thing that we can do with AI and machine learning, and I promise this is like the last thing, and then I'll just go on to final thoughts, is that, and another way that we can use AI with these simulations is to in a multi-scale approach, we can actually zoom in. So when we use these big simulations to identify regions of interest, For example, like the calcium interacting with the mucins or interacting with the mucin and the glycans and the spike protein, we can actually zoom into a smaller area, which we then treat with higher resolution, more accurate simulation. So we can go from molecular mechanics type methods, MD, which I told you about, to sort of quantum mechanical approaches. And NTOS and the folks, Fred Manby and Tom Miller, developed a method that they called OrbNet, which basically was it was a machine learning. It is a machine learning based approach that basically allows you to carry out highly accurate quantum calculations, but it's been trained. So this method has been trained on various DFT methods, so we can apply OrbNet to this sort of more complex system. But it's much faster than if you just ran the DFT method itself. You run OrbNet, you get orders of magnitude more speed, so it's much faster, but you retain basically the accuracy that you get with a higher order calculation, a more rigorous calculation. And again, this is a paper they published in 2022 sort of describing that approach. Why we care about this is that it is true, molecular mechanics, especially for highly charged particles like calcium ions, when we care about these type of interactions, they actually don't do very well. The classical force fields are sort of known to fall short here. So again, this is a way that AI can make our simulations more accurate when and where we need them to be. And so, so in closing, I guess I'd like to say that, sort of facing forward, I think, biology, again, is multi-scale, and we're going to need a diversity of methods and approaches and, to really sort of answer any particular question. I think simulation at this scale, across these scales, gives us a really sort of nice platform to do that. And so this is a bit of a more technical slide, but it's sort of describing what are the different sort of experiments we can do, what are the methods that are being utilized, and then where we ran them. And I think, more and more we're going to see these, sort of complex, heterogeneous composite workflows that utilize a blend of AI and physics-based simulation in order to answer, new questions. And then just also in closing, one of the things that some important notes, which we can talk more about, is that I think one of the biggest challenges right now for biology is really making our data more useful in terms of, we need better biological data production pipelines in terms of like how we actually produce the data, all the things that go into it so that it could be really fully reproducible and also shareable. And the data, of course, needs to be findable and accessible and so forth, but like, there's many challenges with sort of each of these different and changes for different fields, but at each of these different levels, there are important things that need to be worked on. I think we need, of course, better data model integration, where we're going to have significant advances at the interface, as I mentioned, of blending physics-based modeling data and state-of-the-art ML techniques, but we have to bring the right people together to do that. Biologists in general, I think, probably need a little bit of reskilling, and I think that's happening, but we really need to grow interdisciplinary research at the intersection of biology and computing data science. I think that's abundantly clear if we really want to take advantage of the opportunities here. And I think one really nice way to do that, as sort of we showed with SARS-2, but there's many other examples, of really bringing teams together to really go after these domain-driven sort of prototypes right at this space of biological discovery with AI, and oftentimes that means also including the hardware, the cyber infrastructure aspects. Okay, and I will stop talking because we have 10 minutes. Like, we left 10 minutes to chat, but I just want to thank again all the folks who who participated in this effort, and I'll stop sharing, I think.
Thank you so much, Rommie, for a fantastic presentation. Those simulations, those videos are just fascinating to watch. So I appreciate your time. We do have multiple questions. So, and I was also thinking similar to this one question, so I'll start there.
Okay.
So this is along the lines of applications of this work for drug discovery. There's a question here that says, did you try to simulate small molecule drugs which bind and inhibit the spike RBD in either open or the closed forms.
Yes. Okay. Yeah. Right. So I feel like I have to apologize because we also do a lot of work in small molecule drug discovery, but due to some IP reasons, I couldn't talk about some of that work. So then I instead painted this picture, which is I think also really important as sort of the other half of my brain for simulation. So, small molecule discovery against the spike. Yes, we've done it. I'm not really sure. I'm not totally convinced. I don't think it's the best target for small molecule drug discovery for various reasons. It's also, so yes, one can do it. Yes, people have done it, but it's difficult. And it's also difficult. It's been difficult for those of us who, and I'm sure myself and there have been others, I'm sure, who just tried this. Also then to subsequently structurally characterize, these molecules has been in the pockets where we're predicting them to bind is also sort of, it presents also another challenge because you have to at that point probably use cryo-EM, which is fine, but you end up with resolution problems and you have to have a pretty high tight binding molecule that has good solubility and so forth, to actually get that. So if you want to do sort of like structurally enabled discovery against the spike, I think it's one of the more challenging targets. That said, of course, there are other targets, which we've also simulated, but I didn't talk about here. Protease, obviously, MPRO is a huge one where there's a lot of dynamics, occurring, and definitely people are advancing that. We're doing that, for example, together with Ruben Harris and others, where you can get into this whole cycle of generative AI for small molecules and so forth, which is, another interesting, I'm sure probably someone else, maybe Brian Roth will talk about that. I'm not sure when he speaks and as the next speaker in the series.
Thanks, Rommie. We still have multiple questions, so I'll go through them. So this is a follow-up question. Could you comment on the limitations and accuracy reliability of the model using current force field-based MD simulation and possible QM-based simulation for such a large virus model system?
Yes. So, I think a lot of the force fields are certainly a limitation. I think that they are especially challenged when you get to small molecules. I think actually when you get to larger biological structures, like I showed you here in this talk, with the exception of the calcium, because that's, a single ion and highly charged, the force fields actually do a pretty decent job. They're very good at predicting sort of these fundamental biophysical properties and even connecting the timescales of the motions that we see. For example, the opening of the weighted ensemble, I didn't tell you, but using those simulations, which were just carried out with classical force fields, we also predicted the rates of the opening, and it aligned with the second scale timescales that they saw with FRET. So in terms of answering sort of biophysical questions, I think the force fields are actually pretty decent. Where they are more challenged is when you get to small molecules, and you're trying to look at quantities such as binding free energies. You really realize that there are limitations that need to be dealt with. That said, there's this whole initiative called the Open Force Field. There's many folks who are working on this challenge specifically, and they're getting there. There's improvements. There's been steady improvements in that, but making sort of the ability to sort of generalize these parameters to be accurate, but also go across a humongous chemical space, which is really, really large, I think has been one of the central challenges. For QM, I mean, I'm a huge proponent, as I sort of tried to allude to, for multiscaling. I don't think we're not going to answer all the questions we need to for biology using interrogating at one scale. We have to be able to cross the scales and to change the physics when we change the questions that we're asking and sort of how we're interrogating the system. So I think actually, the QM methods for, looking at QM-MM, for example, if you're looking at enzymatic reactions, have advanced, I think, quite handily also. And I think you can have a large system, but then just focus that rigorous calculation down to a region that is, accessible to these methods. And I think OrbNet is another really cool example of that, where you can achieve really sort of high accuracy calculations, but at the same speed that you're basically doing MD. But again, you have to like focus it down to a smaller region and then be sure you're kind of asking the right question for the method you're applying. If I answered the question.
Thanks, Romy. That was excellent.
Okay.
More questions coming in. So here's one. Will reducing humidity in closed air spaces reduce the half-life of virus aerosols?
Yeah, so there's like this famous U curve for humidity, where there's sort of a sweet spot where the virus is weakest, or less, it stays less viable. It's somewhere around like 64% humidity or so. Higher than that and lower than that, and it's not as it's worse for us. So yeah, tweaking humidity is also important. The CO2 was a really interesting finding, also because it actually changes the pH of that aerosol in ways that I think are, to me, were surprising. It actually raises the pH of the aerosol to make it very alkaline. It has they can have pHs of about 12. In any case, that whole area of study, I think, is another place where, speaking of like intersectional areas, bringing aerosol chemists together with biologists and trying to really understand aerobiology and sort of the real fundamental chemical mechanisms around what's happening to these different pathogens or bacteria or whatever, in these microenvironments is absolutely fascinating. And so there are new tools being developed every day that are allowing experimentation in precise ways that are letting researchers sort of answer those types of questions. But yeah, humidity does matter and can be manipulated to affect infectivity.
Awesome. Thanks, Romy. So here's another one. Have you looked into antibody interaction with RBD and try to understand the efficiency of those antibodies? For example, antibodies that recognize masked sites by glycosylation or certain conformational sites.
Absolutely. And in fact, I tried to work it into this talk, but I already had so much stuff. And then I was trying to leave time for discussion. Oh, I could give a whole separate talk. In fact, I usually do on like the antibody stuff. So one is to, of course, rationalize the binding of the different antibodies. One of the most fascinating things to me, actually, which I probably should have included, is how these simulations can be used to predict the exposure of what we call cryptic epitopes that are otherwise either masked by glycan or even buried inside the protein. And we've seen that now for hemagglutinin, for example, which is a glycoprotein of the flu, where we can actually capture these large-scale breathing motions of the HA that reveal one of these cryptic epitopes. And so that's cool. The other thing where we can use simulation, which I think also kind of, to me, and I didn't want to be too provocative in this talk, but like I like AI and I like machine learning. But I also I love learning about these molecules. And I think like to me the idea of like black boxing everything kind of takes out the joy of like really understanding like how these machines work. And one of the things that we did with the spike protein was we worked together with Jason McClelland and a whole bunch of other great collaborators to try to develop a broadly neutralizing immunogen that would use epitopes on what we call S2. So the S1 domain, those RBDs actually like completely come off, and then you're left with just this inside bit of the spike protein, which is called the S2 domain. And the S2 domain has these, it's just much more conserved across the beta coronavirus family. And so it has these potentially broadly neutralizing epitopes. But one of the problems is just like making sure that those epitopes are exposed enough that like you actually can, in the immune repertoire have antibodies that bind to that. Anyway, what we did was actually use simulation to understand how S2 basically opened. And we then used it. So we basically used simulations to understand the dynamics of S2. And then once we understood what was happening, we actually proposed mutations that would stabilize the S2 in conformations that were exposing those epitopes. That work was like just published last week. And I thought was a really cool example of, using simulation to do immunogen design. And it was in some ways almost like the protein design, obviously, like in immunogen design, huge area. David Baker and others are doing that too. A lot of folks, a lot of those methods, you'll see like they make a lot of predictions and they do tons and tons of experiments. And then, the success rate of those experiments, it's relatively low. Maybe they have hundreds of designs and then there's like 2 or 3 that are going to win. But I would say we actually just predicted 2 mutations, and actually they just had to do two experiments, 2 different mutational experiments, and they were able to get this stabilized immunogen, which then allowed them to get the high resolution cryo-EM structure, and it's now, being utilized in downstream experiments. So to me, it's a balance of, do you just do a whole bunch of random experiments, or do you take the time to actually learn something about your target and then use that strategically in a design process? And so it's sort of a balance there. I don't know if that
Rommie,
makes
sounds
sense, but
this work
we're
is
doing
so
that.
exciting. We should bring you back and we can talk
Sorry,
about
I'm
it.
yappy.
No, it
Yeah.
is extremely exciting, and really, so maybe we'll just bring you back. You can talk about it. But hearing from all of this, I'm curious about, it sounds like you're working across a lot of different disciplines, right? Biology, computational work, and so on. So this comes back to one of your points at the end. Is there any guidelines, challenges you can, discuss about being able to work in such an integrative environment? Also, what do you think the community can do to basically help and promote such work, right? Such collaborations.
Yeah, I mean, that's a really good question. I think that first of all, I think, oh gosh, this is like we could have a whole separate panel on what we could do to help this area, right? Because part of it is like the science. I mean, well, it's also, there's academic science, and then there's like, real life science, can I call it? I mean, science of like trying to, I don't know, be more directed in the application. I think communication is a huge part of it. I think it would be good to train students to do more team-oriented science, but then again, that's not necessarily like incentivized in a lot of like PhD studies and stuff. So I think that actually kind of hampers progress in this space. I personally like to see more sort of like some larger projects where it allows different research teams to come together to talk to each other and ask stupid questions, and even just figure out how to communicate. I mean, I have to say, when we were doing this work with the aerosol chemists, it was like they have a whole language and we have a whole language. And even sometimes we share words, like we'll use the word substrate. And if I'm talking to an entomologist, that's like, we think of substrate, we think of this. When you're talking to an aerosol chemist, if you say substrate, they think of something else. And so even just like aligning and the ability to talk, it took us about 1.5 years to really talk, learn how to speak to each other. So I think a lot has to be done there in terms of just sort of like cultivating these connections. And I think it also really helps. There are people, I think, who frankly are good at and open to sort of collaborative science where it's a skill set, and that should be encouraged instead of always kind of downplayed as not as important. But anyway.
Yeah, this is great. Interesting you mentioned the communications part, because a couple of years ago we had this idea of potentially bringing to the Assay Guidance Manual a chapter on communication and guidelines for communication between computational scientists, biologists, and chemists. It was just like like an idea. I'm still thinking about the importance of such a chapter, right?
Yeah.
For
Well, I
someone
think it's
coming into, yeah.
Well, I think it's more important than ever now, especially with AI and AI trying to make inroads into biology and even in, and chemistry also, drug discovery, all these things. It's a challenge.